All right, so glycolysis. We're in metabolism. We are about to go through in the next few chapters the oxidation of energy sources. And we start off the energy source with glucose, the sugar, and we're going to extract energy from that. And when we look at the overall reaction of the 10 steps in glycolysis, we actually net just a very small fraction of the energy that is represented with the glucose at the beginning in the full oxidation of the glucose into CO2 and H2O. Obviously, we have not ended up with CO2 and H2O at this moment in time, so we know we haven't created all of the energy, but we're going to show where the energy has transformed. Now, one thing about glycolysis, and I wanted to point this out, and quite often within metabolism, a lot of our intermediates, a lot of the organic structures are phosphorylated. Okay? That means they have a phosphate group placed onto them. That phosphate group really does a couple things. One, the phosphate traps those intermediates inside the cell. Many of the intermediates will not be transported out of the cell. They may be transported between membranes and organelles inside the cell, but not outside. And so once glucose gets that phosphate on, it's kind of trapped. For it to come off, we have to take off that phosphate, and that's a very controlled enzyme, and that usually only happens in one of our tissue types. Most of the time, once glucose gets inside a cell, it's there for its lifetime. The phosphate groups also make the molecule charged, and there is energy involved in that phosphate. Remember, phosphates always have magnesium roaming around, and so magnesium is always there. Even though we don't articulate it, it is there. And so the first step in the process of glycolysis you've probably seen before, but we're going to look at it a little bit differently. And so when we take a look at organic mechanisms inside biological systems, what we do is we look at the beginning and we look at the end, the reactant and the product, and we see what is different. How many reactants are there? 2, yes. The glucose and the ATP. Now, what you don't see is there's an enzyme as part of this reaction. The enzyme name is hexokinase in most tissue types. There will be a glucokinase in one of the tissue types. We'll take a look at when that is used. And the reaction goes from glucose to glucose 6-phosphate. Now, why do we call it glucose 6-phosphate? It's on the 6th carbon. And so if we number these, all right, there's the 1, there's the 2, there's the 3, there's the 4, 5, and 6 carbons of glucose. Before, it was a hydroxide, and at the end, there is a phosphate that sticks onto the glucose on the 6th position. All right, now, fortunately, on the notes, there is a highlight, but on the exam, there will not be a highlight. What we do is we take inventory, we look at the beginning, at the end, and see what changes. And so here we obviously see there's a hydroxide here, and here there's not a hydroxide. Something different. Now, in the case, they actually probably didn't articulate this as well as they should have. The box here does represent what is transferred. The oxygen is from the original, and so this yellow box should probably have been just on the hydrogen, because that's what's actually substituted. All right? And so I know there's going to be something that happens to this hydroxide in the reaction. All right. There are 2 things that you always have available inside an enzyme. And it is different than what we see in an organic flask. Okay? And so inside an enzyme, you will always have a proton that can be used. All right? And you will always have a base that is used. Now, what does that base and what does that proton represent? All right, if you watch the videos on the organic mechanisms, it was covered, but let me give you an overview. Inside the enzyme, you bind the two reactants that are going to react. Okay, the enzyme does a couple things. It orients the two reactants in a particular three-dimensional placement so that the two things you want reacting are right next to each other. Okay? Secondly, the enzyme will stabilize the transition state, the high energy state by which the reaction has to proceed. Okay? The other thing that the enzyme does is it allows the reaching of equilibrium much faster, so it's a catalyst. The acid is represented by an amino acid in the active site that is charged. It has a proton that is able to be deprotonated, so it can be given. The base, on the other hand, is a amino acid side chain that is in the active site that is deprotonated and able to take a proton. And so if you look at any amino acid, okay, we can determine what the protonation state of the R group, the amine, and the acid, just knowing what the pKa value is and the pH. And we can determine if it's going to be protonated or not. But when we get inside the enzyme, there is what's called a microenvironment inside the active site where things react, where the pH is a little bit different than just the cytosol. And so we can have a little bit difference in the protonated states of our amino acids. All right. Sometimes we'll actually identify which amino acid acts as the base. Sometimes we'll actually tell you which amino acid does the proton, but most of the time I won't. We'll just say there's a base there, there's an acid there. All right. Now, in terms of looking at what happens, we have an acid and a base. Okay, I would say 90% of the time. The first reaction that actually occurs, the first electron movement that actually occurs, is a base is going to take off extra protons. That's usually what we're going to look for. And so if I look at the beginning and the end, there's some change, there's some change. This one has more protons than this one does. That indicates I'm probably going to take it off. Okay? And so I'm going to articulate this just a little bit differently than what it's written. I'm going to put a bond, put a very long bond, and an H, okay, to articulate the hydroxide. All right. The first reaction that occurs is the base will take that proton. Remember, electrons are the ones that are being used to show the mechanism, so the electrons will take that proton. That means you could write, and in an organic reaction, what you would write is these pair of electrons go onto the oxygen. Now, I did a dotted line because that's not what's going to happen here, but having a negative charge on an oxygen, good, bad, okay. Absolutely bad, absolutely good, or okay. More falls in the okay range because oxygens are electronegative, right? Very electronegative, one of the most electronegative things we have. So having a negative charge on an oxygen is okay. It is high in energy, higher than the hydroxide, but it's okay. It's not something that would prevent me from doing a reaction. Now, if you actually were in an organic reaction, you would actually float around as an oxygen negative until you could find your reactant, okay? And because you're not oriented in any space, you're just floating around in the solvent, in the round bottom flask. But because we're in an enzyme, the oxygen that becomes a negative charge is right next to the place where I'm going to react. Now, the ATP has 3 phosphates. How do we designate them differently? What do we call them? Alpha, beta, and gamma. Okay, so here is the alpha, here is the beta, and there is the gamma. All right, alpha, beta, gamma. Sounds kind of like a sorority. All right, where do we attack on the ATP? The alpha, the beta, or the gamma? Okay, how many people say alpha? How many people say beta? How many people say gamma? Ooh, got lots of gamma. How many people said nothing? A lot of people. So, all right, it is the gamma. Okay, so what I would write is this. As the base does its first electron movement, the second movement is attack at the phosphate. Now, every time you attack an atom that has a pi bond, all right, or a double bond, the first electron movement that occurs has to be the double bond must go on to one atom and be a full charge on that atom. So I will kick the electron up onto that oxygen. We just said that oxygen having a negative charge is okay, but that is one of the high energy states. Okay? And it's an excited state. Do things like to stay in an excited state for long? No. Much better it would be if it actually reformed the double bond, and it will. That's much, much more stable. But when I do that, I now have a lot of bonds to phosphate, one too many, and so something has to leave, and in this case, I will break the bond between that phosphorus and the oxygen, and what falls off is and then the ribose and 1, 2 phosphates. What do we call that molecule right here? ADP. And so I know ADP falls off, I transfer 1 phosphate. That lets me know I'm going to attack at the gamma, and I'll kick up, kick down, and the leaving group ADP is a wonderful leaving group. All right. My whole purpose in life in metabolism is to make ATP. That is an energy molecule. Glycolysis is going to make ATP, but right now I'm net -1 ATPs. Okay? All right, first step. Second step is a enzyme. Oh, one more thing I want to talk about the first step. The enzyme, okay? It has a particular class. All right, it is a group of enzymes, and the class of this one is called a kinase. A kinase is used to add a phosphate from ATP. Yes? Yes. Does glucose actually attack the gamma all the time in this enzyme? Because the example in my video showed you could attack any of them. All right. Inside the enzyme, when I bind these 2 particular molecules inside that active site, the hydroxide in the 6th position, right here, is always right next to the gamma. Okay? If by chance I mix glucose and ATP in a round bottom organic flask and heated it up, I would react it at every one of them. The gamma would actually be the most often because it's sterically less hindered. The alpha would be the least one, but if there's no direction, I could attack all 3. But because we're in an enzyme, it is controlled. All right, the next particular reaction in step 2 is phosphoglucoisomerase. Okay, these names are wonderful. It's a glucose with a phosphate, and I'm going to isomerize them. Okay, an isomerase, that's the enzyme class. Now, isomerases, when we look at mechanistic ideas about isomerases, all right, if we actually started to do inventory, and I counted up all the number of carbons, 6 carbons, 6 carbons, all the same. If I added up all of the oxygens, same. If I added up all the protons, exactly the same. All the phosphates, exactly the same. I didn't lose anything and I didn't gain anything. But I did manipulate the structure. Okay, that is indicative of an isomerase. Okay, and so mechanistically, what in the world do we do in terms of going from one structure to the next? An isomerase tends to do this. It's going to deprotonate, it's going to reprotonate. It's going to deprotonate, it's going to reprotonate. It's going to deprotonate and reprotonate until it's done. It'll take off a proton, give it back. Take it off, give it back. Okay? Usually it is driven to one of these structures has just a little bit more stability organically. Okay? And so I will have an equilibrium and I will go to the most stable structural form. All right? And so in this, I'm going to do that deprotonate and protonate. All right? So here is the mechanistic process from glucose 6-phosphate, that G6P, to fructose 6-phosphate, the F6P. All right? I always have a what available? A proton. I will put that proton no particular place, and I always have a base. And I will take and place the base in no particular place. All right? And so, what in the world would you tell me to do first? Deprotonate. I started off because it's all about the base. Okay? And so I will take that proton away because when I look at this structure to this intermediate, there's a proton there, there's no proton there. So I'll take it away. That means the electron pair between the hydrogen and the oxygen is going to go where? It forms the what? The double bond. Okay, when I actually form that double bond, now I have 5 bonds to carbon. That's not good, is it? Okay, so I have to break one bond, and it has to be the leaving group. And the bonds that I could break are the one to hydrogen, the one to a carbon, and the one to oxygen. Which one do I break? The one to oxygen. And I'm going to show this. Okay? Now, that arrow movement, I'm going to show what that represents. You could have written it and said, the negative charge goes onto the oxygen for just a short period of time, and that, stop. There, good. I didn't lose it. And then I would pick that up. But you know, scientists are lazy, so we do the easiest way first, right? And so I can draw 3 arrows, or I can draw 4 arrows. I'm going to go 3, okay? And so I've gotten to the intermediate. Now the intermediate step, there's 2 things about it. One, it goes from the cyclic form to the acyclic form. All right, so I'm opened the ring structure. Okay, but I'm not where I want to be. I really want this oxygen here attached to this carbon there, okay? But there's no reason for it to attach yet, okay? Now, knowing that this is the intermediate, all right, remember, what do I always have to use? A base and a proton, okay? And so I'm going to put the base here and the proton here. And so, remember, this is the first carbon, that is the second carbon. Now let's look at the first and second carbon over here. This is the first carbon and that is the second carbon. Which one has less hydrogen? The first carbon or the second carbon? The second. All right, I have less carbons in the second right here. I have that extra one, it's not there. That indicates where I'm going to start. I'm going to take that away. Now that means the electron pair between the hydrogen and that carbon can go somewhere. Where is it going to go based upon the intermediate? That's the next step. It's going to form the what? The double bond. Okay, I formed the double bond. Now, oh my goodness, the one carbon, when I form that double bond, now has 5 bonds to carbon. Is that good? No. So what happens? Well, one of the bonds to that carbon has to leave, and which bond is broken? That double bond, that pi bond. And I'm going to show it do that. What could I have shown? The double bond could have first what? Got onto the oxygen, be negative charged, and then picked up. Okay? And so, am I where I want to be yet? No, but I'm almost there. Okay? If I look at this intermediate to the final product, okay? All right, there is the one carbon. There is the one carbon, okay? What is the difference between that one carbon and this one carbon? Yeah, there's something moved. There's not a double bond anymore, okay? What is bound to that carbon there that is unseen but known? Hydrogens. How many? Two. So I have two hydrogens, or I have one hydrogen and a blob, the way I wrote it. Okay, two hydrogens on there. It started off in this intermediate with only one, right? Okay. This oxygen here has how many hydrogens? The one in the ring at the product. Right there. How many hydrogens are on it? None. How many there? One. Oh, I found an extra one. And remember, isomerases are take one off, put one on. So I'm going to put my base in no particular place. Okay. And I'm going to put my proton in no particular place. All right. And so I'm going to take that proton and deprotonate it. That leaves the electron pair onto the oxygen, which then attacks. And in the final product, this oxygen is attached to the number 2 carbon. So I attack it at the number 2 carbon. That means this particular electron pair that's in the double bond will pick up the proton, and that will be the second proton on the 1 carbon. I have now made fructose 6-phosphate. Okay. Fructose, have you ever heard of that before? What is fructose? It's a sugar. These are all sugars. But where do you find fructose, the sugar? In fruit. Okay. And so, great. All righty. Reach over and talk to your neighbor and determine what would be the mechanistic steps for this third reaction. Yes, exactly. Look, you don't even have to learn anything new for this one.
It's just I'm so small and I write so big. Mm-hmm. I draw my ears.
Yeah. Yep. All right. Does anybody see something familiar? It looks like step 1. Yeah, it does. Okay. Couple things. Kinase. Okay. Puts it into a certain class, but there's a 1 behind it. What does that make you think of if this is phosphofructokinase version 1? There's more than 1, and so we're probably going to see a 2, maybe 3, maybe 4. I don't know how many, but if there's a 1, there's some others. All right. Does anybody feel good about this mechanism? Yeah, you feel good? Your group felt really strong about it? Good. Well, how good did your group feel? All right. And so, Better when you looked at step 1. So, would your group like to come up and do it for me?
You're going to make me do it? Wait, you want me to draw on
Yes, come on up here. Let's do it. Now, I will do a couple things for you first. Okay, I will actually articulate some things. And so you have a hydrogen that is separated. And then I'm just going to put randomly on this slide, in no particular place, a base for you. Okay?
I appreciate that.
All right.
Oh no.
So,
I'm gonna look at stuff just to make sure I do.
now, I'll tell you this. Sitting at your seat, you can see exactly how to do this. Coming up here to the iPad, everything goes outside your brain. Okay? This is more like when you get to a test, right?
Okay.
All right.
So the base is gonna attack that hydrogen.
Yes.
Hanging off. Whoa.
Whoa. There's a little back. Yeah. There you go.
Oh, it's so sensitive.
It is.
My hands are also shaking, which doesn't help.
Oh. Okay. Ta-da! We got it. All right. So you've taken the proton off. What happens to the electrons between the hydrogen and the oxygen? That sounds great. All right. So you got the words. Show me.
Okay. I'm trying to figure out where I want to draw my arrow. Should I do it from the oxygen just up to?
Well, what you want to do is take the electron pair. And so draw it from the electron pair. Remember.
Okay, so from this bond
Uh-huh.
up to there.
Yes.
Oh,
Oh.
I'm trying to make it so slightly
Yes.
up to there.
It's going to attack the phosphate. Yes.
Phosphate, okay.
Are we allowed to?
This is just not pretty.
Are we allowed?
We're gonna go again. We're gonna go again.
Oh, we're going to go again?
In organic, sometimes we would
I'm
draw
gonna
like
make it
an.
better.
Yeah, I can understand that.
okay?
I can understand that. Yes. I can interpret that.
Oh, it's.
That's fine. That's fine.
shaking, y'all. I'm so
It is
sorry.
going there.
If that's an arrow.
That's an arrow. Okay.
We're gonna use our imagination.
That's good. She's perfectly attacking exactly where it goes. So she hit the phosphate,
There's this indecision.
and
My
so,
hand is like literally shaking.
okay, so now what happens when you hit that phosphate?
It's
What
gonna
is,
swing up onto the oxygen,
Yes.
swing back down, kick off the ADP.
okay, so show me what that might look like.
Oh man.
You can put your hand on the iPad, yes. If you want,
touch it
like,
there?
you can actually put your hand on it.
That's good.
Oh,
That was what I was missing, y'all.
yes.
Okay, so now, do I need to draw that
Okay.
going away?
I'll draw that last one because it's hard.
Yeah.
Okay, so she kicked up, she kicked down, and now we don't have a lot of space, but that, oh. Even with experience. Okay, that pair of electrons will fall onto that oxygen. And so we're breaking the bond between that phosphorus and that oxygen. That pair of electrons goes onto that oxygen for the ADP to fall off. You're very, very good. All right.
My hands weren't drawing straight lines.
Very good. All right. And your
Thanks, friend.
notes.
Helpful, thank you.
All right. Now you get one other responsibility.
Oh no.
Next time someone has to come up, you get to pick who gets to come up.
That's really a lot of power.
It is a lot of power. All right. Step number 4. All right. Step number 4 is called aldolase. Aldolase is a more common name in terms of class. An ACE generally is an enzyme, and ACE usually breaks things. And so this is kind of an interesting reaction. What actually happens to the fructose 1,6 bisphosphate? Yeah, break something in half. What do I break in half? The actual carbon chain, right? All right. Now, oh, interesting. I'm going to go back for just a second. All right. What comes off in this reaction is called ADP. That's adenine what? What's the D stand for? Diphosphate. Okay. All right. This is fructose 1,6 bisphosphate. Okay. What does bis stand for? 2. What does di stand for? 2. What is the difference? There is a difference. The way it's pronounced, yes. The way it's spelt, yes. That, okay. That's perfect. Diphosphate has the 2 phosphates attached together. Okay. Bisphosphate, there are 2 phosphates, but they're separate in the structure. Okay, so that's the difference between a di and a bisphosphate. All right, I'm going to break it into something called dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Okay? And to do that, I have to break a bond, and I have to break it right there. Okay? Now, it's done by the enzyme aldolase. Now, sometimes the enzyme just acts as a base and an acid, okay? But every so often, the enzyme actually becomes covalently attached to the reactant in the middle of the reaction. What it's really going to do is reduce the energy of the transition state. This is one of those cases, okay, in which the enzyme is going to be part of the reaction. Additionally, I cannot break fructose 1,6-bisphosphate in the cyclic state. I actually have to react it in the acyclic or the straight chain. Okay, so I'm going to show you how we get to the straight chained intermediate. To get to the straight chain intermediate, we always react at the what's called the anomeric carbon, or the carbon that was the ketone before we cyclized, the ketone or the aldehyde. And so the base is going to come and take this proton. The electrons will form the double bond, and I will pick up a proton. So before I can do anything, I have to make this the straight chained version. All right? And so fructose 1,6-bisphosphate written in the straight chain breaks into two different intermediates. And I said the enzyme becomes part of the mechanism. So this is linked to on your Moodle page. Here is the reaction. So I have the full organic, the fructose 1,6-bisphosphate, and you see the enzyme. I don't think I can use my pencil here. No, it just moves it up and down. Okay, you see the enzyme, and it's got a nitrogen at the end. That's actually a lysine. The base will deprotonate the lysine. The electrons will attack the carbonyl, and it will kick up. Now, you don't have to write this because you'll be able to print this out off of Moodle. All right? Now, this base here will then deprotonate, cause a double bond, and water falls off. I get a brand new organic functional group called a CHYS base. Okay? That nitrogen has a positive charge on it. Okay? This is what gives us the power to do the reaction that we need to do. All right? Now, we really want to break a bond. We want to break the bond between this carbon and that carbon. 1, 2, 3, and 1, 2, 3. So to do that, a base will pick up a proton. I will form a double bond, a ketone, and a leaving group will be this electron pair here, which then kicks that electron pair up to the nitrogen. Okay? I get half of my molecule off, the glyceraldehyde 3-phosphate. That's half of what I wanted. Here, I get a absolutely neutral intermediate. That means it's a whole lot less energy involved if I have a neutral leaving group. Okay? Now, that's nice, it's beautiful, it's in resonance, and most of it, like 99% of it, is sitting right here. But this doesn't actually proceed to the next reaction. What's going to happen, I couldn't get the arrow exactly right. The electrons goes onto this carbon to create a carbanion. Carbanions are not really great. That's why the distribution is way over here. But this can pick up a proton. Very reactive. Okay? And so if it's 99 and 1 and I react this away, I have 0. Then it's 99 and 1, I react it away, it's 0. And so I can force the reaction little by little to go to completion. Okay? The next thing I have is this part of the compound that is 3 carbons long still attached to the enzyme. Well, I don't need it attached to the enzyme. Now, if you looked up at the top, water fell off to make this intermediate. So I'm now going to bring water back in. Deprotonate the water, the hydroxide attacks. The nitrogen will pick up a proton. I will deprotonate the hydrogen, create a ketone, and the enzyme falls off, and I have created my dihydroxyacetone phosphate. And so up at the top, I got the glyceraldehyde 3-phosphate, and now I have the dihydroxyacetone phosphate. The enzyme is now ready to go through another round. Okay? So the enzyme attacked this particular molecule so that when the breakage occurred, it wasn't just a carbanion falling off. Because a carbanion is so high in energy, it wouldn't proceed. But what did fall off was a neutral compound that only distributed to the carbocation every so often, and it immediately reacted to the next step. And so I needed that enzyme to become covalently linked for it all to work. And now I have 2 small molecules. Step number 5. Okay? What class of enzyme is this? An isomerase. That means the mechanistic steps are what? Deprotonate and then reprotonate. All right, so if I look at the top and I look at the intermediate, I'm going to deprotonate. Now, which one am I going to deprotonate? Well, if I take a look, phosphate is here, nothing happens there. I have a carbon with 2 hydrogens. I have a carbon, 2 hydrogens. Ooh, I have a carbon with a hydroxide. I have a carbon with a ketone. Did I deprotonate there? No, but I did protonate there. Okay, and this top one, I have a hydroxide and 1 proton, and here I have a hydroxide and 2 protons. All right, who would you like to select? I will just randomly, no particular place, give you a base and give you a proton.
It's going to attack here,
Yes.
which is going to put this one here.
Is it going to put it there?
We have no, it won't. It actually,
Okay.
So
good.
it's going to take it here, and it's going to put this on this part, which would be negative, which I'll have a double bond here.
Uh-huh.
So what I'm looking for, so we're going to form our double bond there.
Yes.
And then because that has 5 carbons on it,
Uh-huh.
5 bonds on that carbon, and this is going to go up, and actually, let's do this. Actually, grab the proton.
Look, that's great. That is it. Perfection. Woo. All right. Aha, I was going to say, could I have done this, done that, and done this? Yes, I could have. Why? Why is the green arrows just as right as the blue arrows? The hydrogens are equivalent. Okay? Now, mechanistically, if I really wanted to know if it was the top or that side one, you could get really creative and synthesize an intermediate that has a proton and a deuteride. And you could actually test to see where the deuteride goes to actually prove which one. But because they are actually equivalent, you probably get a 50-50 chance of either one of those. Now, that doesn't finish the reaction, does it? I'm in an intermediate state, right? Okay. So what in the world do I have to do to finish this up? This enzyme does what? Its mechanism is to do what? Take a proton off and put a proton on. All right. Noah, pick the next volunteer. Oh, and the precedent has been set. The precedent has been set. It has to be a female. It has to be wait a minute. No, it has to be a female. The precedent was set. Oh. What? Okay, I am just going to write randomly anywhere on this chart where a base and an acid might be. Okay? Now, all right. It's scary when you come up here. Okay? I got scared just standing up. Okay. All right. Now, when you're up here, what in the world do you do other than stop breathing and start crying? Okay? What would you want to do to figure out what the electron movement is going to be?
Phone a friend.
Phone a friend, yes, you'd love to do that. And you could probably do that on homework, but not on the test. Right. So what would you give her an advice before she actually even picks up the pencil?
Are you deprotonating or reprotonating?
Ah, yeah. What? Yeah, see what changes. And so, can you see what changes? What changes? Tell me.
Got a little bond up here.
Mm-hmm.
Or where you lose the little bond there.
Okay, yes. And where, if you're going to have to put an extra proton, where do you think it's going to go?
An extra proton?
Yeah, you see, up here, that's going to be lost, right?
Right.
But I have to take off one and put one on.
This guy.
Okay, so that's right. All right, now, do you got a better idea what you're going to do?
I think so.
Okay.
Maybe.
Maybe? Let's go.
Let's try it.
Let's try it.
All right, Ellie, let's see how hard this is.
Actually, touch your hand on the thing. I was trying to hold it up, and that's why I was shaking.
Take this guy.
Ta-da! Yeah, she deprotonated.
Okay.
Now that pair of electrons between the hydrogen and the oxygen goes.
To this carbon.
Does it go on the carbon?
Maybe not.
No, no. Look and see what you see.
Oh. It's a double bond.
Yes. Well, now you've gone really back.
Uh-oh.
Yes.
In here.
There. Okay.
Oh, this is too many.
That's too many. Yes.
So this guy comes and grabs this guy.
Yes. Perfect. Absolutely perfect. Woo! All right. Big picture for me. Why in the world did we do this slide? What did we do? We changed what to what? We changed dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Wait a minute. Step back. What did we make in the last step? Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So at this point in time, I've taken glucose and converted it into what? 2 molecules of glyceraldehyde 3-phosphate. If we didn't do that, glycolysis would have had to have 2 separate pathways, one to deal with dihydroxyacetone phosphate and one to deal with glyceraldehyde 3-phosphate. But because we took the dihydroxyacetone phosphate and made it glyceraldehyde 3-phosphate, we have simplified how many enzymes we need to fully degrade the glycolysis. All right? And so we've simplified the problem. Now, from this point onward, every reaction that we see is represented by 2 reactions. All right? Okay. And so, we've got a new organic concept introduced into this particular reaction. Okay? Here is the concept. I have something called NAD+. This is an electron carrier. Okay? The electron carrier likes to carry electrons. Okay? And it will become NADH, but what it's going to do is it's going to react with a hydride, which will react with this to create the NADH. This means anytime you see an NAD+, you get to invoke an entirely new leaving group, which would probably be the worst leaving group you would ever write in organic chemistry. And that's a hydride. Hydrides are not good leaving groups. But if in an enzyme you have an electron acceptor, the enzyme will facilitate a hydride to leave. Okay? And so anytime I see NAD+, I know hydrides can be a leaving group. All right? Now, this particular reaction is going to involve a redox reaction or a hydride moving. The enzyme class that does that is called a dehydrogenase. Dehydrogenases can allow hydrides to move and must have an electron acceptor. This is one of the many electron acceptors that we're going to encounter. Now, remember, the enzymes can either just act as acids and bases in the active site or sometimes become covalently linked. This is an example where the reaction, the enzyme actually becomes covalently linked. Okay. Inside the enzyme, we have an NAD plus and we have a cysteine as a active site, as an amino acid in the active site, and it is deprotonated. So it's a negative sulfur. Okay? And so the molecule, the glyceraldehyde 3-phosphate, will come in, and the sulfur will act as a strong nucleophile, and it attacks the electrophile, which is the carbon of a carbonyl, because it's slightly positively charged. Now, this is probably the only time this book actually gets this right. When it attacks the carbon, the electrons kick up onto the oxygen in an intermediate state. It's the high energy state. The electrons don't like the high energy state. They will kick back down to form the double bond, and in this movement, the hydride is a leaving group. If ever you wrote that as a hydride leaving group in an organic reaction on an organic test, most likely, like 99.9% of the time, would be wrong. Okay, but this can happen in enzymes. All right, that's great. I've gotten rid of the hydrogen, which is what I need to do to get to the product, but now I've got this molecule still attached to the enzyme. What will come in, well, first the NAD diffuses out of the active site, another NAD plus comes in to reset, and a phosphate comes in. Now, phosphate has negative charges, so it can act as a nucleophile, and here's where the artist gets it wrong. It attacks, and obviously it knows that you know it kicks up, it kicks down, and I just lost it. And the sulfur is the leaving group, and now I have 1,3-bisphosphoglycerate. Okay. The enzyme here is a dehydrogenase because the electron pair moved. We had a redox reaction. All right. One other thing about this reaction. I have just placed a phosphate onto the intermediate. Every other time that this has happened, I've actually had to use ATP and break it down. I've had to use energy. Here I added a phosphate to the mechanism, to the organic molecule, without using ATP. It is a very powerful reaction that nets us the potential to gain energy without investing energy. All right? Next reaction, the 1,3-bisphosphoglycerate will become 3-phosphoglycerate. Okay, the reactant is ADP. The product is ATP. Okay, so I always have an acid and I always have a base in these reactions, right? But this is one of the times that the base doesn't start it. What is happening here? A phosphate goes from to what? From the starting material to what? It goes to ADP, the diphosphate, to make ATP. Well, that's weird. Okay, and so what I'm going to do is, any of those negative charges can be a nucleophile, and it is going to attack that phosphate. Kick up, kick down, and what happens is the leaving group is an acid. Now, what's important about that particular group? I drew it that one way. Is that the only way to draw that group? No. What is that end group doing, that functional group? It's resonating. What is that functional group that is outlined in red? That's arrowed. Yeah, it's a carboxylic acid that's been deprotonated. Now, this is cool. I've used 2 kinases way at the beginning, used 2 ATPs. I've just netted 2 ATPs. Wait a minute. How did I net 2 ATPs? I only show 1. 2 molecules of that starting material. Okay. So I'm net 0 in energy. All right. So that was a really big step. I gained energy. I gained the ATP back. Group 8, okay, it's a mutase. It's a whole different kind of enzyme. What functionally happened here? What did you see? The phosphate is on what position in the beginning? The 3rd, right? And it ends up in the 2 position. Well, how in the world did that happen? Okay. Magic, no. God did it. Yes, he designed an enzyme for it, okay? Now, what is interesting about this particular reaction? The phosphate in the 3rd position is not the phosphate in the 2nd, okay? Now, you might think that the oxygen up here could attack here, and that be the leaving group, and it's an internal attack. It's too constrained for that to actually happen. So the enzyme is going to be part of this. And there's something special about this enzyme. Here's the enzyme. Here's an amino acid chain, an amino acid side chain in the enzyme itself. What does this R group represent? Which of the 20 amino acids? You got a 1 in 20 chance of getting this right. Not arginine, but arginine is in that same class, histidine. Okay? It's a histidine molecule, and the histidine comes in with a phosphate. All right? And so, the histidine's sitting there with a phosphate. I bring in my starting material, and I will create an intermediate that is a diphosphorylated intermediate by deprotonate the hydroxide in the 2nd position, attacking the phosphate, kick up, kick down, and in this case, I will even have a stabilizing effect because I will take that positive charge, and when the phosphate leaves, it'll become a neutral molecule. All right? Now, when the reactants are in the phosphorylated state, when they are in the phosphorylated state, and this hydroxide is the hydroxide, this R group is actually directly right here. As soon as the phosphate comes off, the enzyme goes under a conformational shift. Okay, what does that mean? It changes shape. Okay? And it changes shape, and this is where a two-dimensional piece of paper doesn't really do a good thing. It puts this molecule, this side chain, right here next to this phosphate. So it changes its position. It once was sitting here, and now it is sitting there. Okay? So the next part of the reaction is I reform that double bond. This attacks. I kick up. I kick down. And this will pick up a proton to become the hydroxide. So it comes in with one phosphate, and it leaves with a totally separate phosphate in a totally different position. A mutase, we're going to see several of them, usually goes through this intermediate that has two phosphates on them, and that intermediate never leaves the active site, and it is a way in which we transfer from one to the other. It looks like the same phosphate, but it's not. All right. We're going to do the last, another enzyme that has a generalized name called enolase. And quite frankly, this is actually the first reaction you ever learned in organic chemistry. Yes?
I do have a question.
Okay.
If you said identify as mutase, would you tell us it's mutase, or do we have to actually look at it okay, it changed here?
Ooh, that sounds like a wonderful question. Where I just show you the And is it an isomerase? Is it a... That sounds like a great question. Let me think about that for a while. Yeah. You would actually know it if you could look. Yeah, that sounds like a great question. Yes?
Can you explain again why it has that phosphate instead of getting back to the other?
Okay, so initially when it's bound, the structure, actually phosphates actually constrain structure inside an enzyme. And so with the phosphate on, the whole protein actually puts it right about here, right next to this hydroxide. When the phosphate is off, that allows freedom of movement and the protein shifts its structure, putting the histidine now close to this one. And then when it picks up the phosphate, it's going to shift back over into the other position. And so there's two shapes for this, and depends upon whether it's phosphorylated or dephosphorylated. All right, you learned this particular mechanism in organic chemistry. Most of the time, it's one of the first ones that you learn. It's like this. You have, in the book, a C, a C, and they usually write just bonds. And then they usually write a hydrogen and a hydroxide, okay? And if you do it with a base catalyzed reaction, the base will take that hydrogen, the electrons will kick in to form the double bond, and a hydroxide falls off to form that. That's what's written in an organic book. That's exactly what's happening in this particular reaction, except I gave you R groups. Some of them are big R groups, but it's just R's. And so sometimes when you look at this, it's hidden. The reaction is hidden behind. It's a very simple reaction. What we really need to focus is on not everything in the whole molecule, just where the reaction occurs. So don't get scared by there's a lot of stuff there. Identify what changes the small little space. And so in this case, I am going to have, instead of a hydroxide, I'm going to have just a base and just a proton, and I will take off this hydrogen, because when I did inventory, this carbon that had that phosphate on it doesn't have a hydrogen. And then this carbon is now absent a hydroxide. And so I form the double bond, and the hydride picks up a proton to become water. I get some really strange named molecule called phosphoenol pyruvate. We don't like saying that, so we abbreviate PEP, phosphoenol pyruvate. And we are almost done. The last reaction is, kind of familiar before. ADP goes to ATP. I'm going to allow the electrons to attack the phosphate. I'm going to kick up. I'm going to kick down. Okay? And this has just a little bit more movement, because I could put those electrons right onto that oxygen, but that's still a little bit charged. So I'm going to put those electrons to form the double bond, which is that double bond there. Too many bonds to a carbon, so this carbon is going to pick up that proton, and I'm going to be left with pyruvate. Okay? And so, the overall reaction here. I told you had to know the overall reaction. Here is just the overall reaction you need to know. All right? Glucose plus 2 NAD pluses plus 2 ADPs plus 2 pre-Is yield 2 NADHs, 2 pyruvates, 2 ATPs, 2 waters, and 2 protons. All right, I want you to know this. Here is all the potential energy that we started with. It's in the bonds of glucose. Okay? Where is the energy now at the end of this? Okay, definitely the ATP. Unfortunately, too often we make you think that that's the only energy source. Okay? There's other energies that are still left. Where is the other part of the energy? What? Yes. The NADH is a pool of energy. We're going to see that that pool of energy can be converted into ATP, although it's going to be converted into another pool of energy before it gets to ATP. There is another place where energy is left. In pyruvate. I still have carbon bonds. I still have things I can break down, or essentially more carbons that I can oxidize. That's really what I'm doing. And so I've distributed the energy that was all in the glucose molecule into a little bit in ATP, a little bit into the NADH, those electron pairs that are in that carrier, and most of the energy still left in pyruvate. Okay. In a process like glycolysis, 10 steps. Most of the time, what we do is we regulate this whole process, and we can regulate it as little as one single enzyme that will either start or stop this whole series of reactions. Glycolysis is a little bit more complex because I can take intermediates from several different places in glycolysis, and so we have three main places in which regulation occurs. The largest regulation occurs at the phosphofructokinase version 1 step. Okay? And in actuality, these are also the three irreversible steps, because if you take a look at all of the reactions I had, there were equilibriums. They could go back and forth. But these were the steps in which either ATP was made or broken. And so these are so high in energy that they are not in equilibrium. But these are the places in which we regulate. All right, the first enzyme I talked about to you was hexokinase. As soon as sugar, the glucose, gets inside a cell, all right, we have hexokinase in most of the cells. Glucokinase, though, is an enzyme that is only in liver. Now, liver and muscle are going to be the compare-contrast organs, okay, because they use glucose in two separate ways. All right? Now, what does this graph mean to you? Okay? If you looked at enzymatics before, it should mean something, but usually at this moment in time, if I give you this graph, it means absolutely nothing. Okay? All right. What in the world? What are we measuring in the two axes? The reaction rate versus concentration. Glucose is the reactant, so as I get more reactant, the reaction rate is the production of glucose 6-phosphate. Okay? And so as we increase the concentration of the substrate, what happens to the activity, or how much product is formed? If you increase the concentration of the reactant, you get more production of product, right? Are they the same? Not at all. Okay. All right. Does anybody know, or has anybody ever seen Km? Okay. Michaelis-Menten constant. It's a constant that is derived from the action of an enzyme. Can anybody remember way back when and give me an approximation of what Km means? Oh, I love it. 1/2 Vmax. We got down to an equation. Well, what in the world does 1/2 Vmax mean? What's Vmax? 1/2 of the maximum velocity. Yes, that is exactly how we determine Km. Okay, but what does Km actually mean? Well, Vmax, or the maximal velocity, is reached when all of the active sites are filled completely. That's as fast as the enzyme can go. So what do you think happens when you're at 1/2 Vmax or Km? Half the active sites. That's exactly it. All right. Now, if you talk to an enzymatic biochemist, They will totally disagree with me, but most people think about it this way. It's the concentration at which half the active sites become full, so it's kind of like a good measure of how well it binds. And so which of the two, hexokinase or glucokinase, which one has a higher affinity for glucose? Hexokinase. The lower amount of concentration reached Vmax or half Vmax faster. We always compare at the halfway mark. All right. The Km for hexokinase is 0.1 millimolar. The Km for glucokinase is 10 millimolar. And the normal concentration of blood glucose is about 4.4 millimolar. So what does that mean? At normal concentrations of glucose, hexokinase is working at what percentage? Basically 100%. But glucokinase doesn't get to even half at normal concentrations of blood glucose. When does it get to half? When it gets way up here, okay, 10 millimolars. How in the world do you get to 10 millimolar concentrations of glucose in your bloodstream? Yeah, you can get it there if you're diabetic, and if it's at 10 all the time, that is really, really bad. But you can also get it if you eat a Snickers bar, okay? But if your metabolism's working right, you have high concentrations of glucose for very short periods of time because the insulin is sent out, glucose is absorbed in all the tissues around, okay? And in liver, only when it gets really, really high will liver start to take glucose out of the bloodstream because glucose intent is to give glucose in the bloodstream to keep it at the right level. But it will store it when you see it's high, and then it will feed it to the muscles when it's low, or feed it to the brain, really. Okay? And so there are different versions. Usually we talk about hexokinase, which is in the muscle, and glucokinase, which is in the liver. Does the class officially go to 3:05? Okay. All right, so I need to start winding it down. All right. So the isozymes 1 through 3, which are the ones that are called hexokinase, are in the muscle. They tend to take every single bit of glucose and trap it inside the cell for muscle to use. Glucokinase will only trap glucose inside the liver when it's very high in concentration. And it's regulated by how well it binds to glucose. Now, how in the world does the body understand that glucose is there and that we're in the well-fed state? We know well-fed state, insulin comes out and is secreted. This is how it's recognized. Glucose gets transported by the GLUT2 transporter. It goes into the cells in the pancreatic cells where insulin is actually secreted. Glucose goes through glycolysis to make pyruvate. Pyruvate will take 2 more chapters to get to ATP. ATP will go up. ATP inhibits a potassium channel. It closes it. It becomes depolarized and opens up a calcium channel. The calcium will influx in and it will go to vesicles that have insulin inside and cause those membranes to fuse to the plasma membrane, and we will exocytose insulin into the bloodstream, and it tells every other cell in the body, hey, there's lots of glucose around, I have lots of energy, I have lots of starting material, and it will start up a lot of different processes for lots of different tissues. And it's all because glucose gets transformed through glycolysis in the pancreas. Okay. All right. And so, hexokinase 4, or the glucokinase, is in the liver. Liver really doesn't want to do glycolysis. Its hexokinase version 4, the glucokinase, actually hides in the nucleus most of the time. All right? Only when glucose gets into the cell at relatively high concentrations will it cause the regulatory protein to dissociate and hexokinase gets into the cell. Okay? And hexokinase will phosphorylate it. What the liver wants to do is store this glucose, send it off to storage molecule. If by chance the storage molecule gets complete, it will start breaking it down through glycolysis. But the cell doesn't want to do that. When I get the fructose 6-phosphate, the steps of glycolysis, when that concentration goes up, it goes and says, hey, regulatory protein, grab this, put it away, stop. And so the liver has extra catch to make sure we don't use it for glycolysis. We want to trap glucose, but only for storage, not for breaking down. All right. So, what do we need to finish in this chapter? All right. If you would go to the first quiz, and I think the chapter numbers are wrong, but this is the first quiz. Okay, so what does that say? Draw the mechanism of glycolysis, including any enzymes that make covalent bonds with the substance during the reaction. There were a couple of them that actually became part of the reaction. Aldolase was one of those. Okay? And so I want you to draw out the entire mechanism. Okay? If you would like to do that on ChemSketch, you can. No one's ever attempted that. Okay? If you would like to do it on a sheet of paper like this and you draw it all out, perfectly fine. Okay? I will post up this video very shortly to help you complete that. Okay? But I want you to think through the process, because you can sit and listen to it and watch it and, oh, it makes sense. But when you have to do it, that's when you have to think about it. Okay?